Okay, we're recording. So welcome everyone. This lecture is going to be about in vivo imaging. It's going to be a primer on in vivo imaging. We have another session next week. The session for next week is supposed to be a hands-on. Initially, this was, of course, planned to be in a classroom setting. So it'll probably consist of a lecture / hands-on. What we'll go over an analysis of images that are collected in vivo, and but we'll also have a component of lectures still. Today I'm going to be talking about in vivo imaging in a very broad sense, and I have a few disclaimers before I start. Let me just move your photos to somewhere else. Okay. So for the first one is that this is going to be a very practical presentation, and I kind of took the cue from Luke to discuss things that you don't learn in textbooks, like for example glue, right? That he was talking about in the last lecture. All of these little things are important. Also, I'll try to highlight a few things that you may not have seen. Everybody sees the big papers that come out in Nature and in Nature Methods and Nature Neuroscience. At some point, I'll try to highlight things that may be emerging or technologies that may not be very explored yet. At some points, the scholarship may be a little spotty, so I'm not always citing original papers. I've surely skipped influential and useful work. And I'm going to be, mostly this lecture is about two-photon microscopy in mice. So you need to be aware of that. So there's plenty of people doing zebrafish, flies, and everything. I wanted to include some of that. Basically, I thought that I would just talk mostly about mice, and as an example, because it's mostly meant to give you an introduction to the possibilities of the system and of these systems that are available. I'm not going to be talking, there's a lot of very good work that unfortunately I've completely overlooked, so I just even removed it because I wouldn't do it much justice. Many of the examples are my own work and that of colleagues, just because those are examples that I can think of immediately. They're not necessarily the best examples in the field. Some images that I may show you may not have the correct attribution. Okay, with that said, the first thing that I'm talking, the title of the lecture is imaging from fiber photometry to miniscopes. And in vivo imaging, fiber photometry is actually not really an imaging technique. It doesn't require an image. It doesn't require the formation of an image. You don't have actually. It doesn't require a bitmap. So fiber photometry is a bit of a simpler approach to take, for example, GCamp, which you guys have talked about last week. So calcium sensors, for example, you can do this in freely moving animals, and you just insert the fiber and you excite GCamp, and you collect the fluorescence from the GCamp to a sensor that's outside, so the mouse is tethered, but only by this fiber, and you record from multiple sites, and you can record multiple colors. This doesn't even require imaging, and this is a place I chose to start to say, whenever you try to do, if you ever want to do imaging recordings or photonic recordings, start simple. Never do anything more complex than what you actually need. And for a lot of stuff, just recording bulk signals is great, and it's enough. I mean, I'm going to give you a little bit of this. I think is kind of one of the state-of-the-art papers of fiber photometry. This is a really cool work by the Helmchen lab. So this is the system they use. It's basically a camera, which is a CMOS sensor. It's pretty much the same sensor that you have at the back of your cell phones. Then you have an excitation light source, which is this laser. You have a dichroic to separate the excitation light from the emission light, the blue excitation light from the green emission light of GCamp. And then you couple both the excitation and the emission to this fiber bundle, and it's basically a fiber array. And you see that the arrays that you have, all these little fibers, they have different lengths. So you can just basically stick this into the brain, as you see here in panel D, right? And this is really cool work because they record simultaneously from a bunch of different sites. So each one of these fibers is in a different point in the brain. And what you record is, if you have GCamp, for example, in these areas, you can get all of these channels and record bulk calcium signals from different areas and see which cells are activated during any kind of behavior, right? And this is probably an example of a very complex kind of state-of-the-art fiber photometry system. So you know, you can do this. You already talked about electrophysiology. You talked about all of these arrays and silicon probes and tetrodes for imaging, for recording activity in vivo. So why is, and you have even these other options, which are recording bulk signal from GCamp, where you can see the variation in the activity of cells during behavior in mice that are almost freely moving, they're still tethered. So why is imaging useful at all? And I think that it gets down to a few points, and I'm going to go and give a few examples of these. Imaging is useful when you have population codes versus just looking at variation of rate encoding versus not rate encoding. Mind you, you can get this kind of information also with silicon probes to a certain extent. But imaging can be very useful in some circumstances where silicon probes struggle, or any other type of electrophysiological measurement struggle. And I'm going to show an example in a little bit. So you also know which structures, subcellular structures you're recording from, meaning you can record from, and I'm going to give you a few examples during this presentation, you can record from individual synapses in vivo. You can see the topographical structure, the location of the cells as you record. You can do this, of course, in vivo. That's the whole point of this whole course. You can do this long term, and you can track the same cells and be absolutely certain that you're tracking the same cells over time. And in all of this in a minimally invasive way. Remember, everything is invasive. Everything that we do, almost every experiment is invasive. But with imaging, very often you don't even need to pierce through the dura. You can do these recordings with just doing a craniotomy. I'm going to show you that you can even do it without doing a craniotomy. You don't even remove the bone. And you have the potential for recording from a huge number of neurons. So why is it useful? So one example, I gave you this, I told you about the idea of recording morphology, fine morphology. This is something that you really can only do in vivo with imaging. I'm giving you an example from my own work. In my lab, we study this population of adult-born neurons in the dentate gyrus. And these neurons are developing so they start from the stem cell stage, and they extend these dendrites. And you see here, we're actually able to sparsely label them. So these are reconstructions. So these are not the actual images, but we take images of these cells and reconstruct them. And so these cells are labeled with GFP, and you can track them over time. And we track them for about 60 days as their dendrites are growing. And you can tell which, and this is kind of the key here, you can tell which branches are added between one imaging session and the next, and which branches are removed. And this is something, this ability to know the before and the after and to track the same cell over time, this is really unique to imaging, and you can't really do this in any other in other ways. Now, with activity, there's also a big use for imaging. For example, depending on which areas I don't know if this may be a problem. So I have a few videos, and I don't know if we'll have trouble with the rates of Zoom versus the rate of the video. So I don't know how this shows up. Do you see stuff flashing?
Yeah.
So we do see activity. The thing that I found out about Zoom is that anything less than, say, 10 hertz is fine, but more than that, it's going to mess up the video.
Okay, 10 hertz should be fine. This is an example of imaging of cells in CA1, and you see that there's a lot of cells flashing. And the problem is, so, people have studied place fields in these cells in CA1 with electrodes for a very long time. The problem is when you're studying cells in the dentate gyrus, and I'm showing you a video. This video is not necessarily darker, it may be a little bit darker, but what's happening here, if you look carefully, and hopefully you'll see this in your Zoom presentation, is that this is an extreme example of a population coding, meaning there's only very few cells that are responding during this movie. Very few cells are active. And this is actually a very, very packed area. So, if you can see my cursor here, this is the granule layer of the dentate, sorry, this is CA1, but this is the granule layer of the dentate gyrus. You see that this is a highly dense area with a lot of neurons. And here you have activity level comparisons at any given time in the hippocampus. You compare, for example, entorhinal cortex, area CA3, CA1, subiculum, and you see here that in the dentate gyrus, less than 0.5% of all cells are active. Now, this poses a problem for electrophysiology. Why is that? Guys, does anyone want to tell me? Why is it a problem for electrophysiology when you have only a few cells that are here and there, spread a little bit over? Yeah. Someone is saying something?
Because I think if you put the electrode in, it seems like you're not recording from any neurons, but they are there, they're just not active. So you can't detect them.
That's exactly what the problem is. When you have very sparse signals, you stick it, traditionally the way that we've done recordings in the brain is to stick a wire into the brain and you see what kind of responses you get. And the problem here is that if you do that in the dentate, most of the time you hear crickets. It's absolutely nothing, right? So you need to sample. So with electrophysiology, activity is very sparse. It's also relatively, with a lot of techniques, unless you do patch clamp recordings and you fill the cells, very often it's very hard to identify because it's such a dense area. It's very hard to identify which cells you recorded from, and there's actually a heterogeneity of populations here. So it's very hard. So for the longest time, we've struggled to get good activity information about the dentate. And people have resorted to immediate early genes. And that's why you're probably familiar with the whole engram papers and the forgetting and the papers with the artificial memories. So these papers study immediate early genes because for a very long time, that was the only tool that you had. So, in this particular area, for example, you want to record the activity of hundreds, even thousands of cells because it's very sparse. If you want to get meaningful data, you need to sample a lot of them. You want to identify the cells genetically. You can do this also, for example, with optotagging. There's other ways of doing it, but with here you can have a co-label. For example, you can have parvalbumin cells express a red dye. You can do this during a behavior task, and this is really something very unique to imaging techniques. So hopefully I've convinced you in a couple of examples that there are things that are unique to imaging that give you in certain circumstances, they can give you very distinct advantages. Not all circumstances, sometimes it's not the best technique to use. So now, why is it in vivo imaging difficult? Why has it been a challenge? Why is there so much? So we know that you've probably heard there's all of this intense research on in vivo imaging techniques over the last 10 to 20 years. And part of the issue is that the brain is not clear. It's a difficult substance, a difficult tissue to image through. And the brain is not clear. And what happens when you have photons, so light crossing through the brain, is that these photons get scattered by electric clouds in the matter of the brain, in the tissue. It's not an optically clear substance. So both the excitation and the emitted light are scattered, but really the scattered of the emission is usually a lot more limited. And absorption in some cases, so can also play a role, but it's not really that the brain is opaque. It's really more the scattering. And what do I mean by this? So this is work so this is work actually by, Adrian had this idea. So this is work that I did during my postdoc with Adrian Cheng in the lab of Carlos Portera at UCLA. And we were imaging a bead, a single fluorescent bead, and we're putting different slices of tissue, of brain tissue, on top of this bead. And you see here, you start with 0 microns of tissue, so we're imaging just a plain bead. This is the axial distance. So this is Z. I'm pointing. You cannot see me pointing. So this is axial. This is XY, and you're seeing a single bead from the side. And this is kind of what a single bead looks like. It doesn't look round. There's an elongation of the point stretch function. We'll talk a little bit about that. That's very typical of imaging systems. And then if you start putting tissue, just a slice of tissue between the objective and the bead, you start seeing this progressive distortion of your image. So what you get is this broadening of the point that you initially had. And this happens because you have scattering, and scattering works a little bit like these pachinko games, in this Japanese pachinko games, where you have one of these little balls and then a bunch of pegs, and the balls will bounce off in all sorts of different directions, and you will not get a straight line. So the photons will actually end up, by some of them ending up, if you have enough of pegs, so if you have enough of the scattering tissue, the photons will end up kind of far away from the place that they would normally end up in. So how do you solve this? How can you image under these circumstances? How can you image through tissue? Well, one thing is clear, is that whereas when you were imaging, for example, if you're imaging cells in a dish, it's a very thin preparation, you probably can get away with just using a CCD or a CMOS, so just basically a camera. Here, image forming methods like a CCD, like the camera on your phone, are not a very good way to go. And the point is, when you have all of this spread, so the photons you're collecting can come, you never know where this photon came from, then there's no way that you get a good image with a CCD. Although now there are techniques to do that, and we'll go into that into the miniscopes a little bit later. But what you do is you use laser scanning microscopy. So what does this mean? Instead of having a CCD or a CMOS, which is basically like the camera on your phone, or a photo camera, where you have pixels, you here take a completely different approach. You have a single pixel detector, which is a PMT that I'm going to introduce in a little bit. So you have one single pixel, and you form the image in a different way. So you go and you probe individually different points in your sample, and then you collect all the light that comes from there. And the light can come from anywhere, but you know which area you excited. So you no longer use a bitmap. You compose a bitmap individually, and you scan one single point at a time. So That's good, because now you know that you're scanning, if you're scanning this point and you get the photon hits the microscope, hits the sensor way off axis, you know that you excited this point, and you're good, because you know this is the point that I don't need to care about where the photon came in. I'll just grab as many photons as possible. And in fact, when you do in vivo two photon imaging, sometimes if you have a really strong sample and you go in the dark, you go under the microscope and you see a little green glow. If you have a mouse that expresses a lot of GFP, you see the green glow. That green glow, you can even put a sensor there on the side of the mouse. It doesn't even need to be through the objective. The objective is only needed to excite here. How you detect doesn't matter. You can detect from the sides, you can detect whatever green light there is in there. So the second way that you increase your resolution and you deal with having this thick sample of tissue is you do optical sectioning. And there's different ways of doing optical sectioning. For different reasons, multiphoton microscopy is probably the best way of doing optical sectioning in tissue, in deep tissue. And by optical sectioning, we mean when you eliminate a specimen, you get this cone of light, right? So you get your focal point and you have this cone of light through your sample. And you want to really image one of these points at a time. So you don't want to collect fluorescence from areas that are outside of your focal point. And in vivo two-photon microscopy allows you to do that. And it works like this. So two-photon absorption is a much lower probability event than single-photon absorption. So when you excite, for example, GFP with blue light of the microscope, pretty much you're exciting all the GFP in this cone. But by using two-photon microscopy, because the probability is much lower, it falls quadratically from the beam, from your focal point, which is the point where you have the most concentration of photons, right? Because your light is mostly concentrated in this focal point. So it's really a matter of probability. You want to have as many photons at a single spot at the same time. And this is your focal spot is the only point where this happens. So the probability of absorption is much lower, and you need a lot of photons in this one point at a single time. And by the way, two-photon microscopy was, two-photon absorption and the physics, the photochemistry of two-photon absorption was first postulated by Maria Goeppert-Meyer during her dissertation thesis in Göttingen. And she actually postulated that fluorescent molecules would be able to absorb 2 photons instead of 1. So what does she mean by 2 photons? You absorb, whereas in a normal fluorescent molecule, and I think you guys covered fluorescence with Luke in the previous lectures, right? So usually you have the excitation of a fluorescent molecule from the ground state to the excited state, and then you emit. For example, with GFP, you excite with the blue, emit with the green. Here, what you do is you excite with photons of approximately half the energy. So because they have half the energy, you need 2 of them. And if you can extend this to 3-photon microscopy, you need, approximately a third of the energy. Energy is not exact. And you need 3 photons instead of 1. Now the problem is that these photons need to align along the dipole, the excitation dipole of your fluorophore. So basically here we're playing a numbers game. You do this optical sectioning because you only have excitation at your focal point, because it's the only spot where you actually have enough photons.
Tiago, I have a question.
Yeah.
So in the previous slide, what is the volumetric empirically driven data that we know that? What is the volumetric size of that spot that you're talking about?
Well, that will depend, and it will depend on a lot of things. It will depend on your on your power. So it falls quadratically with the z-square, but it will depend on your point spread function of your imaging system, because what you'll need to do is create this volume, which will be the smallest, you know, ideally the smallest volume, that the smallest points that if you're diffraction limited, that your objective can image. So it will be, you know, the kind of like the point spread function volume approximately.
Is this, for example, the microscope you have in your lab, is this smaller than usually the size of a cell or a dendrite?
I'm going to talk I'm going to talk about this. No, yes, this is definitely smaller than the size of a cell, but it depends. So you can have, it depends on the objective. It depends on your imaging system. So you are limited here by your imaging system. So not necessarily the two photon. So what matters here is kind of your focal point, the size of your focal point, and even the amount of laser that you're pumping into the system. Because remember, it falls quadratically. So if you have very little laser power, then you will also get a smaller volume excited. But we'll go into that into a second. But yes, in vivo two-photon microscopy allows you to go sub-synapse resolution, or synaptic resolution approximately. Now, when Maria Goeppert-Meyer did her dissertation, she said, you know, she proposed, yeah, I proposed this theory that you can actually excite a fluorescent molecule with two photons, not just one. But she had no way to test it. And the reason why she had no way to test it is because back in the 30s or in the 20s, whenever she did her dissertation, it was actually, we didn't have a single light source that had the power, the amount of power. Remember, it's a low probability event, so you need a lot of photons at the same place at the same time, aligned with the dipole of this fluorescent molecule. And we just didn't have the kind of tools to produce that kind of light. So this changed, and it changed, I believe, in the 80s. We started having light, and people finally were able to verify this theoretical work and say, yeah, there is this two-photon excitation works. And in the late 80s, early 90s, the first lasers that were practical to use this in a microscopy, in microscopy, using this technique in microscopy, the first lasers were made available. And these are titanium sapphire lasers. These are relatively clunky and complex. They have a pump laser, they have a lasing medium, they have different, it's actually more, it's a laser cavity with a pump laser attached, and they have moving parts inside. And they're relatively, so you always need to tune them, and they're relatively big, expensive, and complex. They're being completely replaced now. And in the future, what you're gonna have is different lasers with different single wavelengths, most of them based on fiber laser systems. But as of now, this is still the most prevailing technology. And these lasers produce very short, and we're talking about 100 femtoseconds. Here you have a pulse is about 200 femtoseconds. But these are very short pulses of light, and they produce these every 12 nanoseconds, usually around 80 megahertz. And the peak power is very, very high. So you don't fry the brain, because for most of the time, remember, you have these 100 femtoseconds, but that's a lot less than 12 nanoseconds. So for most of the time, the laser is actually off, right? So the duty cycle is extremely low, but you have every now and then this huge burst of energy hitting your sample. And that's what allows you, so the peak power is very high. The average power is about 2 watts, but you don't even use that to image. But this is what allows you to excite your sample with two-photon excitation. Now, very short pulses also pose problems. One of them is that a very short pulse these lasers are tunable, and you say, I'm using, for example, 910 nanometers, which is the wavelength that you use to image GFP. So you're imaging 910. That's what you're using to excite. But actually, your laser is not only 910 nanometers. You actually have more frequencies than just 910. It's not a single color laser. That's why you have these pulses here, which are actually, they're femtosecond pulses, but they actually have a range of wavelengths. And this comes from bandwidth theorem. It comes from the fact that the width, the full width of at a half maximum of a pulse, and the frequency components of a pulse, this product is a constant. So if you want to have very, very short pulses, you'll have broader pulses with more wavelengths. There's no way of making a very, very short pulse that is composed only of a single wavelength. So these lasers are very different from, say, continuous wave lasers, like a 48 argon, that only pump out 488 nanometer light. And sometimes, if you have dispersive media, you actually need to compensate for the dispersion, because the problem is the velocity of light. So you guys know what the velocity of light is, right? So I'll need to bounce this off just to make sure that you guys are following me, because this part can be a little tricky to get. So
2.98 into 10 to the power 8 milliseconds.
Okay, and what is that in
meters
glass?
per second.
That is perfectly correct, but that's in vacuum.
In vacuum, it's less in glass.
Right, but it's not only that. Is that it's different. It depends on the wavelength.
Yes.
So this is the nuance here. So that means that red and blue wavelengths will not arrive at the same time to your sample. So you're trying to put all of this power into your sample, and you know it's very important to have all of these photons at the same time, except that, you know, your red photons are going to arrive later than your blue photons. So the way you solve that is to actually give some of your photons a head start when they leave the laser. And this is called group velocity dispersion compensation. And some microscopes need this in order to operate. So I'm going to start by talking a little bit about the actual parts that go into a two-photon microscope. First, I guess we have the femtosecond laser that I just talked about. And this produces all of these little pulses of light. And then you want to scan your sample. Remember, we're not imaging all of, we're not illuminating the whole sample at the same time. We're illuminating one point at a time, and we're scanning it. And we do this with XY scan mirrors. And this is really just a set of two mirrors and I'm going to show, I have a photo in there. Two mirrors that are you're controlling, and they're basically oscillating back and forth, and they scan your point along your image to make to produce an XY image. Then you have a set of lenses. You have a dichroic mirror to separate the emission light from excitation light. You get your excitation light into the objective, into the sample, and then you collect the fluorescence back and add a PMT. And as I said, your collection of fluorescence, you want to collect as much fluorescence as possible. You can even collect fluorescence from the side of the mouse. It doesn't really matter. So a little bit about the anatomy of this. So most lasers you'll find do not actually have a dial or anything to tell you, oh, you know, you're going to use more power or less power. So all of these systems need to have a control. So the lasers are always on off. There's nothing else. So all laser systems will need some sort of power control system, and there's different ways of doing it. Usually they involve some sort of polarization control. So you have a half wave plate to polarize align polarization of your laser light, and then you have a polarization beam splitter here, which basically either allows a certain polarization, say this direction, and then the other polarization is going into a beam dump, and is really just a beam dump. So you're just dumping whatever part of the beam you don't need, whatever laser power you don't need, you just set it to the side and you put it against, it can be a cinder block or whatever. There's other ways of doing this. For example, one of them is with a device called a Pockels cell that you'll see very often in many optical systems. And Pockels cells are basically electro-optical modulators, and they basically select light based on polarization too, and can do that on a very fast time scale. So you have your laser, you have your beam splitter. Here you have a part of your laser that you don't need because otherwise you'd fry your mouse. You have a shutter usually, and then you have a set of mirrors, and you pump this laser into the microscope. But before you pump it into the microscope, there's another consideration, which usually you have a beam expander. Why is that? Well, I'm going to be talking a little bit about objectives in a second, but you want to actually backfill your objective of your microscope, meaning you want to have, in order to have the smallest possible point in an objective, because what's on one side of the objective is the Fourier transform of whatever you put in, you actually want to have as flat illumination as possible there. And we're going to go back to that. But that's why you usually need to have some sort of beam expander in your system. So, and this is where then you pump your light into the microscope. And this is actually where your scanning mirrors are. And one thing, a lot of courses that do two-photon microscopy courses, they actually have you disassemble and reassemble your own microscope, and you realize that it's actually a very simple system. So these are galvanometric XY scanning mirrors. So basically they respond to a current. You have a command current, and they oscillate back and forth. And and we're going to talk a little bit more about those. And then you have a scanning lens. You have the slide. This is just between bright field and two-photon, and then you pump your laser into your objective, and then you have your mouse here at the end. So laser scanning. So there's fundamentally two different ways. Well, there's more than two different ways. I'm actually not talking about acousto-optical deflectors, which are still a different way of scanning. So I'm only talking about mirrors. There's other techniques that do not even involve mirrors, and they're very fast, and they're great for accessing random points in your sample at any time. These are good for scanning, for sequential scanning. And they're basically a Y and an X scan. And with this, you can compose a whole XY image. And one of them is a fast horizontal scan, and then your lines are a little bit slower, and these respond to a control signal that produces a certain amount of current. So usually you pump in a control voltage, and the voltage gets converted into a current that basically makes these mirrors oscillate. Now, they are limited in their speed, and this is a very important factor, because this is something to understand, is that imaging methods, when you're trying to image activity, the methods that we have are generally much slower than electrophysiology. They're much slower than action potentials. And this is something to keep in mind. Even though your objective very often is to quantify action potentials and the firing of cells, in reality, you're going at a much slower speed. So at this point, I have a mystery figure here. So these are images of in vivo two-photon images of neurons labeled with GCamp. Do you guys see anything strange about this? Anyone? Is there anything strange about these images? Do you notice here, this cell, for example?
The horizontal lines.
Yes, exactly. So any idea of what's happening here? So what's happening is that you're scanning, and there's different ways of scanning. So you actually pump a control voltage, and usually you can do, most of the time you do a sawtooth wave. So you do something like this. So you scan a line, you go back to the beginning, you scan the next line, you go back to the beginning, you scan the next line. But there's also bidirectional scanning, which is faster. So you're moving the mirror like this, and you're scanning on both the way forward and on the flyback of the mirror. The problem is that this is not as repeatable as setting the mirror back to 0 all the time. So because your mirror has a little bit trouble aligning the backwards motion with the forward motion, you get these artifacts, and you need to basically correct them in the computer. So this is a very common artifact sometimes that you see in images when you acquire them with scanning systems. So this is faster to scan both on the forward and the back motion of the mirror, but sometimes it can lead to alignment artifacts, which is what you see here. All right, so you're limited. You want to go as fast as your calcium indicators, and usually we're talking about calcium indicators as you guys heard last Tuesday. You want to go as fast as your calcium indicators can go, and in order to increase your speed, you sometimes use these resonant galvanometers. So these are they go much faster. They go at 5 to 8 kilohertz, and they allow you to do frame rates of, say, 30 hertz instead of 5 hertz. The problem is that they never stop, and they can only go one speed because they're resonant. So they're set with a resonant. They have a resonant frequency that they oscillate at, and they can't do anything else. So so these are used in any system that needs to go fast. So for calcium imaging, these are usually the preferred modes. The problem is because you can't really adjust the speed, sometimes you run into problems with your dwell time, your pixel dwell time, meaning you collect too few photons because you're going so fast that you actually don't illuminate a single point for long enough to collect enough photons. So this everything will look a little bit like an underexposed photograph, like a photograph you took in a club at night and your phone didn't really do a very good job. So that's why when you have these, whenever you take your phone and you have these night shot modes, basically what happens is that your phone is averaging a lot of frames and going slower, basically. And this is what you need to do very often with this kind of imaging. Arvind, do you have a question? No, you just raise your hand. No. So one problem with this, though, is that these are resonant mirrors, and their motion is sinusoidal. So you have something like this. What's the problem with this? Can anyone guess what the problem with this kind of motion is? With this kind of scanning, sinusoidal scanning? Think about the linearity, how fast you're going.
At no point the mirrors are stopping at a single point, so they would.
They never do. So not during the scan. You always collect when the mirrors are in motion.
And the amount of time spent on each angle is very different.
Exactly. That's exactly the point. So this poses a problem because you want to be here in this area, right? In the area that is kind of approximately linear. The problem is when you start getting to this point, what happens is that your mirrors start moving really, really slow. And remember, usually you're pumping a lot of power in here because your dwell time in every pixel is very short. And suddenly they almost grind to a halt and then they start going back. But the problem is you're applying a lot of power at the edges of your image. So what you usually need to do is to have this thing called the Pockle cell that I talked about, this electro-optical modulator, and blank your laser when you're at the edges of your window. And the reason is you'll actually see the grains start melting on the sides because you're burning the sides. And so you really want to only illuminate your sample here and not in the other time when the laser is going much slower, because if it's going much slower, it spends a lot more time and it really will burn your sample.
Sorry, by blank, do you just mean like turn it off?
Turn it off, yeah.
Okay.
So you actually have these very fast electro-optical modulators. And this is just a polarization base. It's a crystal that selects for, it's a set of crystals that select for polarization, and they basically divert the laser into a different direction and out of your microscope. But they're activated by a voltage. So they can be, oh, I lost the drawing that I had. But they can be synced with the sine wave to kind of, whenever the sine wave reaches a certain level, this thing triggers a voltage and it'll just blank out the laser. The laser will be going elsewhere, usually into a beam dump.
Okay, cool.
Now, objectives. So when you're imaging, now the objective is pretty much the most important part, is the most complex part of your imaging system. Modern objectives are quite complex, and we're seeing actually a resurgence of people designing, now it's really funny. There's a lot of labs in biology even designing their own optics. Has become a thing because we're demanding so much more from our imaging methods. But the commercial objectives are quite complex, and you want objectives that are suited for multiphoton imaging, meaning they need to be good they need to have good transmission at the wavelengths that you use. And you need to be careful, because if they don't have good transmission, it means that they have good absorption. And what does that mean? What's the risk with that?
It'll heat up the lenses.
Exactly, yeah. Anything that absorbs is bad. It's not bad because it means that you lose laser power, but it also is especially bad because they'll blast through your lenses and you completely ruin them. So you need to be careful. Not all objectives can be put under a 2-photon microscope. So they need to have good near-infrared transmission. And they need to have good visible transmission, because usually if you're imaging, for example, GFP, that's what you need, right? Now, the most important parameters of an objective for 2-photon are the numerical aperture, the low magnification, the magnification, but you actually want to have the numerical aperture. That's the most important thing. And you need a good working distance. Working distance is basically how far the area that you're imaging, how far it is physically from the objective. Now, you need the working distance because now we're more and more imaging deep areas of the cortex, deep structures within the brain. We're imaging the hippocampus. So for example, when those images that I showed you of the dentate gyres, they are actually about 2.5 millimeters below the surface of the cortex, which means we need to have at least a 3 millimeter objective, working distance objective, in order to reach that. And now there's all of these specialty objectives. Now, one important point is that your objective has this back window. I should have actually made this a little bit more clear, but your point here will be the Fourier transform of whatever you put in. And you know that if you in the simple unidimensional single dimensional space, the Fourier transform of a single point is a flat line, right? The direct delta function. So when you go to two-dimensional space, you want to have kind of an absolutely flat illumination of the back of the objective in order to get the smallest possible point of the other side in your sample, because you want your focal point to be the absolute smallest possible. Now, the problem with that is that lasers are Gaussian. You know, you don't have anything that is just a flat, uniform illumination. So you kind of expand your laser beam, and that's why you need this beam expander, which is usually just a little magnifying, a little telescope, 2 lenses. The problem with that is that it's a little bit of a trade-off, because if you expand it too much, you completely lose power, because your beam will be very broad, and it'll start hitting the size of the microscope and the metal around the lens, right? And if you don't expand it enough, then the size of the point that you'll excite will be bigger and bigger and bigger, and you lose your resolution. So what is your resolution? What is the minimum? And this is, Arvind, this is kind of related to what you were asking. What's the minimum? The resolution is defined as a minimum distance between 2 points on a specimen that allows them to be discerned and you be able to differentiate between these in a diffraction limited system. And this is an approximate equation, by the way, and there's different equations depending on what kind of beams and what kind of systems you use. But it was a limit put forward by this guy Abe, who worked for the Zeiss microscope company back in the 1800s, I think. And he postulated that, you know, whatever the minimum distance that you're able to resolve with a microscope, it's going to be lambda, which is your excitation wavelength or your emission wavelength, divided by 2 times the numerical aperture. And the numerical aperture is actually a measure of the catchment angle of your objective, how much light it actually captures. So if you imagine that a point, you have a single GFP molecule in space, and it will emit photons in every different directions along a sphere. The numerical aperture really captures how much of the sphere surface is captured by the objective. How much of that goes into your microscope? Because your GFP molecule is emitting everywhere. It emits down into the mouse. It emits up to your microscope. It emits to the sides. And sometimes when you go next to your mouse, you see it. So it's emitting everywhere in all sorts of different directions. But you want to know how much of that how much light are you actually capturing with your objective? And the numerical aperture is a measure of that. And it's dependent on the refractive index of the glass. And it's dependent on, you know, which will usually be the refractive index. It'll be matched to your medium, which is usually water for in vivo to photon microscopy, which is 1.3 approximately, 1.33 or so. And it's dependent on the sign of this new angle, which is the angle here at the full aperture, angular aperture of your objective. So, but you know, you don't need to know that, but you just the most important concept, if there's one thing that I want you to take from this lecture is that your magnification does not define how good your resolution is. Your numerical aperture is how good your resolution is, meaning you can have a 20x lens, objective lens, that is actually resolving smaller things than a 63x lens or a 40x lens. So your numerical aperture is what defines how good your objective is. And if in doubt, there's even more. If you have a 40x objective and a 20x objective, and they're both the same numerical aperture, let's say they're both 0.8 numerical aperture, which one do you think will have the best resolution? The 40x or the 20x? Or will they have the same resolution?
They'll have the same resolution.
Theoretically, yes. But empirically, when you go and measure, and for different reasons that have to do with matching the aperture of the objective with the aperture of the microscope, actually lower magnification objectives achieve a higher resolution than higher magnification objectives, which is counterintuitive, but that is the point. So take-home message: your magnification doesn't mean that you're better at discerning small things. Your numerical aperture is what matters. And of course, the wavelength that you use to image. Now, a little bit on point spread functions. I don't know how many of you are familiar with the concept of point spread
I have
function.
a question about that.
Yeah.
So does a smaller magnification objective then have a larger field of view, for example? Or I guess
No.
what difference does magnification make?
Yes,
then
it has a, yes, okay, sorry. The other way around. Yes, it does have a larger field of view, but there's actually a paper about that, and I think I have it in a couple of slides. But empirically, what they found is that if you can make full your angular, I guess the angular exit from the objective, if it makes full use of the aperture of your imaging system, of your microscope after the objective, then you'll actually yield you better results in terms of resolution than if you have an objective that has more limited angular aperture at the end of the objective, at the back end of the objective. There's actually a paper about that I'll actually, I think that's probably one of the few papers that I'll go back to you on that.
So is a 40x zoom, what it's doing is it's bending the light more. Therefore, the exit angle that you have is tinier than that you would
that's
have had
basically what it is.
Okay.
You're not making full use of the, yeah.
Okay.
So point spread functions. So point spread function is basically, in an ideal world, with an ideal imaging system, a point would appear as a point. However, in a real imaging system, a point is not a point. A point is a blob. And it's usually elongated along the axial, the axial, not axial axis, around your objective axis, or the z-axis. So a point does not translate, a diffraction limited point does not translate as a sphere, as a perfect sphere. You have more like a blob that is elongated along the z. Characterizing this is the function that characterizes this is your point spread function, which, as the name indicates, is what a point actually turns out to be when you image it with your imaging system. And it will not be a point, it will be a blob. And your measured image is basically a convolution of whatever your object is with your point spread function. So in theory, you can actually deconvolve that, in theory, and improve your resolution by deconvolving your image. In practice, this yields mixed results. And it's always a good thing to measure your detection point spread function, because that is the true measure of what resolution you're going to be having. And you can do this with beads. Beads, fluorescent beads, are basically these little microspheres that you can buy from Invitrogen or whatever it is called now. It used to be molecular probes, now I think it's thermo. And you can have them smaller than the resolution of your microscope. So you know that they're a diffraction limited point, and then you scan them and you see what kind of shape do you get out of them. And from there, you can calculate your point spread function and see what a single point, a diffraction limited point, what it translates to. Now, a few notes on image size and sampling. So you guys have been through the sampling lectures and the digitization and part of the lecture. And a few notes on that. The zoom feature on your microscope is basically a digital zoom. That means it changes the sampling rate. In some systems, it changes the angle that your mirrors are actually scanning. So it changes how many sampling points you do on your specimen. And when you increase your zoom, you'll get to a point that you have empty magnification. Just like when you increase your zoom on your phone, if you have one of these older phones that only have one lens, you know that if you zoom in, you're not actually getting a better image. You know, it starts getting really pixelated. And it's the same here, meaning your useful zoom is basically 2 to 4 times the maximum theoretical resolution. That is 2 times the frequency. It's the equivalent from the sampling theorem, right, that you guys have covered. Now, commercial confocal systems, for example, you can still get useful data even if you sample up to 8 times the maximum theoretical resolution. But you know, with in vivo phone, there's no point. And then you want to talk also about how fast you need to go, right? I mean, this stuff is taking up hard drive, both you know the resolution of your system and how many images how fast you go with your imaging. Now, how fast you go, very often you'll be imaging calcium, and how fast you go. And I had a slide about this. I don't know what happened to it because we actually did I was patching cells, and patching cells with a really, really fast system goes much faster than 30 hertz. You can actually get data in the pretty much 100 hertz. And then what we did is we had the information about the firing from the patch clamp, and then we had we had our calcium traces, and we tried to deconvolve them. So we tried to get back to the patch clamp, to the electrophysiology data from the calcium data. And what you find, and then you start averaging your data to simulate what it would be like to go slower and slower and slower and slower, meaning at what point do you go too slow to get to actually be able to reproduce the firing of the cell? Because there will be a point where you can no longer reproduce the firing, you lose information. And we find that up to 25 hertz is useful. Everything, and this was done with caveat emptor, is done for fluorophore, but fluorophore is a small molecule calcium indicator, but it's still generally faster than most GCamps out there. So up to 24 hertz, you have useful information. After 25 hertz, you don't really gain anything. So that's kind of I think it's a relatively easy rule of thumb. But whether you can even go 25 hertz, you may not be able to, because it really depends on your sample, because you may be getting more noise than anything else. Yes, Arvind?
Yeah, I was wondering, what is the firing rates of the cells that you're looking at? If it's beyond a certain point, if it's a fast-spiking neuron, then it's difficult.
Those are different effects. That has to do with the saturation of your sensor, of your calcium sensor.
Right. Okay. This
But
is assuming the calcium sensor is perfectly fine.
That is true.
Okay.
But actually here, what you start seeing is the opposite, is that if you only have a single action potential, sometimes you miss it. If you have 2 action potentials, you always catch it. But if you have the singlet, you can miss a lot of those if you're going too slow. So image detection.
Tiago,
Yeah.
I just wanted, so we're actually about an hour in, and now might be a good time soon if we could take a break if there's a natural stopping point.
I have 2 slides, and then it's a good break point. So 1 or 2 minutes.
Okay,
So,
yeah, just wanted to.
Yeah, so your detectors in vivo phone and most imaging systems that are laser scanning are going to be photomultiplier tubes. And these are pretty much the quietest detectors available with the least noise. And they basically work with a photoelectric effect. So you have your incoming photon, and you have a photocathode, which is basically a metallic surface. And the photoelectric effect means that you will get an electron yanked out of your metal by your photon, and you'll be accelerated over a voltage. That's why your PMTs usually have a voltage. If you ever use a confocal, you set a voltage, right? And basically you have all of these different stages, and over each, because you have a voltage accelerating the electrons, you're adding kinetic energy to these electrodes, and every time they hit one of these diodes, they'll have more energy, so they'll yank out more and more and more and more electrodes. In this way, you get an amplification of your signal. And this is how you were able to detect really, really low, signals. Now, there's a bunch of things to keep in mind for PMTs. I'm not going to go too much into detail, but you want to have usually a quantitative efficiency, meaning how many photons actually manage to yank out an electron from this detector to start with, is probably less than 30%. And there's a lot of factors to keep in mind: quantum efficiency, gain, the spectrum, and the speed. The speed can be really important if you're going in a really, really fast sampling. Now, this is a really important paper, I think, is by Martin Noheim, who worked with Social Park in Paris. And this is basically parameters influencing depth of imaging. And I think this is where this guy talks about matching the angular acceptance of the microscope to that of the objective can result in a gain of about 3-fold in the collection efficiency at large depths. So a low magnification 20x high numerical aperture objective further increases fluorescence collection by a factor of 10 compared to a standard 60 to 63x. And this paper actually goes over this and a bunch more. So this is one of the most highly recommended papers, I think, for understanding efficiencies if you're ever wanting to image deep tissues with a two-photon microscope. And this is where I stop. Resources, a few resources. The microscope manufacturers have fantastic websites. They're mostly the same text. They're written by someone in Florida. I never understood who writes these things, but basically they all have different branded, basically they're encyclopedias of microscopy. This RP Photonics, I think they're like this photonics consulting business, but they also have a pretty good encyclopedia. The Data Foundation has this neat little list of brain imaging technologies with a short description. It's almost like a dictionary, and it's actually really useful because there's so many of them now. And then there's this Thor Labs and the UCLA Miniscope website. So I recommend all of these, and now it's a good time to go on a break, 5 minutes. Is that right? Okay, so it's 11:40, so we'll be back at around 11:45. Good.
Hey, Luke, I have a question if you don't mind.
Yeah, sure. What?
I was wondering, so when you, for example, record at 10 hertz temporal resolution of say 10 cells and using GCamp, the latest GCamp and A2 photon microscopy, I wonder if the signal that you get is equivalent to, for example, the summation of spiking activity or the field activity in that cell over the 10 hertz resolution. That is, every 100 millisecond, you're taking an average of the signal that has happened there, events that are happened there, or is it a vision over that tiny instance of moment, whether in that every 100 millisecond you have a recording? What is it? Is it an integration? And if so, how much of a temporal integration is it?
Yeah, so it's not temporal integration. What it is just that the beam hits that point in the sample at one small sample, piece in time, and then it samples that at just that time. But the thing is that if you think about GCamp as a low pass filter, you just have to oversample the frequencies of that low pass filter. So GCamp itself is an integrator of the calcium signal. So you see what I'm saying? So in that sense, you're still under. You just have to oversample your indicator.
So if you
that's why.
If you oversample if the indicator at any instant, if it takes say 5 milliseconds, say 10 milliseconds is the delay of one single, is the convolution window size of the GCamp indicator, then you want at least greater than that resolution to.
Right, but it's much longer than that.
Okay.
Yeah, it's much longer.
Yeah.
What is that time?
There's different GCamps to some of them. I don't know what the fastest one is. The slowest one is, like the GCamp 6S is, or 7S with like 150 or something. Tiago, do you know?
I don't remember. I actually have a slide with that toward the end that I'll probably not get to. But let me see.
But that's one of the reasons why voltage imaging is so hard, is because the events are so short that you have to sample
Exactly.
the kilohertz frequencies basically, which is impossible, almost impossible with scanning imaging.
Yeah, so the single action potential for GCamp 7 slow. Maybe this time scale is not very useful, but anyway. You see the decay, but even the rise time.
Hold on, that is 0.5 seconds. So it's more than a few seconds.
Yeah, the decay, yeah.
Yeah, okay. Wow.
Yeah, but if you have more action potentials, you actually start to see a compounding effect here until it saturates.
Huh.
So if you think about this from a sampling perspective, you have to sample fast enough that you're oversampling the kinetics of your indicator, but then you also have to choose. But then if you think about what's the firing rate, how often do cells fire action potentials, then in a sense, if you want to capture that, you have to choose your GCamp kinetics to match, the expected rate of action potentials. If you had cells that fired very sparsely, you'd probably want like the slowest GCamp you can get to make it easiest to image.
Because your rise time is not that different. Okay, I think we're ready to start again. Okay, so now I'm gonna switch gears and talk. So you saw that this presentation is very much about in vivo two-photon imaging, and there's been no mention of anything else other than a mouse. So I'm going to move on to talk a little bit about the preparations, the different preparations that you can use to image activity in vivo, activity or morphology in vivo. So we're not only talking about activity, I guess. So you can image through the skull, you can image through a thin skull, or you can do a craniotomy where you remove a part of the skull and you implant a glass window, or you can do a craniotomy plus implant a device that can be a light guide, or it can be just a hollow cannula, something that allows you to basically image deeper into the tissue. First one, imaging through intact skull. This is actually possible, and it's been, and people usually do this with 3-photon excitation. So 3-photon is just, I'm not going to be talking too much about it, but it's an even more lower probability event than 2-photon. You can go with even higher, into your sample with even higher wavelength light that will result in less scattering. So this is by the lab of Chris Su, who's one of the big experts. He actually kind of inherited Webb's old lab at Cornell, which is where 2-photon microscopy started. And this technique is relatively new. It's not very widely used, but you can actually polish the bone and use index matching glue. And you can do calcium imaging in layer 2, 3 at 8 frames per second. And this is how it looks like. So you actually keep the bone completely intact, right? You remove a part of the skin. You still have a cover slip here, but you have this index mat. You polish the bone and you have this index matching glue. And the results are actually quite surprising. I mean, he managed to image it all the way down to 500 microns. And this is basically his point spread function. So the full width at half maximum of a point, both in the axial distance and lateral. And this is calcium imaging traces done through in layer 2, 3, done through the intact skull. So this is really, really cool that you can do this and not even go through the skull. Another method is skull thinning. And the skull is thin with a drill and then a blade, and it becomes, usually around 30 microns, 20 microns thin. You need to be very careful when you do this, because as you use abrasion basically to thin the skull, and you can overheat the area that is underneath the skull. So you can actually cause damage by doing this due to the heating. And you can usually image immediately after thinning. So there's no need to wait for inflammation to subside, because if you do it properly, there's very little inflammation. And it can result in reduced inflammation. And a lot of people that work, for example, with microglia imaging, they tend to prefer this just because it's less invasive. But then the bone will regrow and limits the duration of experiments. Usually in 5 days, it's gone. And you can rethin it again, but then there's a limited amount of times you can do that. But you can't image as deep, usually only about 300 microns. And there was a big controversy around 2007 between, I guess, the labs of Wenbiao Gan at NYU and the Svoboda lab. Because Svoboda and other researchers used this craniotomy method where they should remove part of the brain entirely, part of the skull entirely. And Wenbiao Gan kind of claimed that, differences that they saw in spine turnover. So spine is one of the things that spines, in the turnover of spines, how persistent spines are over time, is one of the things that people have really used 2-photon microscopy for. So they claimed that there was a different turnover, and they claimed, this is because your windows are really bad because you have all of this inflammation. And then there was a rebuttal to this, saying, no, we don't see this. I don't think that went anywhere. So basically, the most commonly used method is still the craniotomy. Most craniotomies are about 2 to 5 millimeters in diameter. The tool used to do this is air-powered dental drills. They're the best. They're very cost-effective. You buy them off eBay. You can get one for about $150 around there, with all the pedal control and everything. So you have a pedal, you control this thing. You need to be very precise. You want to prep this around your area. So this is like kind of a plug for Luke's glue slides. Use Optibond in the area, not where you're going to drill, but everywhere else. And I usually like to use the Optibond and cure it. I don't like to use the Optibond. I always feel that the blue light will lead to inflammation if you do it after you've removed the skull. It may be voodoo, but that's. And then you don't really want to dwell as you drill, because you heat up the tissue. And ideally your drill should never breach the bone. So you actually thinning the bone, and then you're removing this plate of bone. Critical, avoid midline and sutures. Most of this applies to, unless you know what you're doing. And then you implant a cortical window, implant just a glass cover slip with dental cement. An important tip, when you remove the cranium, your brain will start swelling. You can get an edema even, because your brain will start swelling, come out of and start pushing out of the bone. So you want to use dexamethasone, so a steroid anti-inflammatory drug, to prevent brain swelling. You want to usually do this before you start drilling. Don't use carprofen or ibuprofen or any NSAIDs before drilling, because otherwise you get a lot of bleeding, because they're also blood thinners. Now, if possible, it's good to combine your viral injection if you're using a virus, and most people do, in order to image calcium. Combine your viral injection using window surgery into one single surgery. And the reason for that is whenever you're injecting the virus and then you cover the mouse back up, you'll get scar tissue. And when you're trying to be very careful about keeping your window clean and not have any blood, but then you remove the bone plate that you just drilled, and you have the scar tissue formation, so you actually grab a bunch of the scar and you make a big mess. So when you inject a virus, the best results are pulled capillaries and using microinjectors. You image acutely immediately after surgery, or you wait about 2 weeks at least after surgery when the inflammation subsides, because you will get some inflammation. This is an example of a surgery. So this is a relatively good craniotomy. You see there's a little bit of blood here, but I don't know if you see here, in all of these images, there's actually a capillary, a pulled glass capillary injecting into the brain. And you should see a little dimple here. You see where the capillary, this is the capillary, and there's a dimple, and this is where you're doing the injection. So you need to be really careful about avoiding all the areas with blood. People say, trauma surgeons say that all bleeding stops, but not necessarily in the best way. And like if you have any bleeding here, it will most likely continue after you cover it with a glass cover slip, which is the last step here. You see, here it's a full implanted cortical window. You have the dental cement here to make it adhere to the skull. And you see actually here there was a little bit of an air bubble. This is not a problem, it'll diffuse out. It won't be there next time when you look at it. And this is what a clean preparation should look like. And a clean preparation can last easily 3 months. Now, not all of the window lasted for 3 months. You see here, this slightly speckled area, this is bone regrowth. So over time, you will get a little bit of bone regrowth, but a lot of areas are still very clean. So this particular window is over 90 days old. So, and this is all great if you want to image a cortex, right? But often you don't want to image a cortex. You want to image, the hypothalamus or whatever. So then you need to implant some sort of a light guide or a hollow structure, a tunnel, basically. You need to tunnel in to go to the area of the brain that you want to image. And one way of doing this is with thin lenses of fibers. If you're around 0.5 millimeters is what people use, and people use a lot of grid lenses. I'm going to talk a little bit about that in a second. You insert these into the brain with your stereotaxic as if you're doing an injection, and you don't actually need to remove tissue. However, if you're using stuff that is a little bit bigger and requires either freckles or cannulas or sleeves, and this is sometimes very helpful for reusing expensive lenses, then you do need to remove some of the tissue. And this is, of course, true whenever you require tissue removal. So large lenses or cannulas. And the advantages of using this cannulas and large lenses are obviously higher increased field of view. But in the case of the cannulas, if you have a larger space where you're actually imaging, you also get higher resolution. And when you do these, this is kind of an extreme, a relatively big example of, so this is an example of making space in the brain. So removing tissue, you do it with a surgery where you have, so this is an aspiration surgery. And here, this is a corpus callosum, by the way. This is the hippocampus down here. And when you saw, so you start gently aspirating this tissue. You need to be irrigating all the time. But this is how these surgeries look like. You see that here, that's the ventricle, next to the hippocampus. And you create this space, and then you implant your cannula. One system that we use in the lab. Actually, we don't no longer use this. This is going all the way to the hippocampal fissure. We now use pretty much the same, but much shorter, that we implant right above the hippocampus. We don't actually touch the hippocampus. And this is a technique pioneered by Adi Mizrahi when he was in the CATS lab at Duke. And it's also used by the Tank lab, by Dan Dombeck and others, to image the hippocampus, and became a very popular approach. It gives you a huge field of view. You can actually image pretty much all CA3, CA1, dentate gyrus, hilus. You can image everything with this one single implant. And that's why, even though it removes a relatively large part, you do a 3mm craniotomy. Just what it gives you in terms of access to the hippocampus is really terrific. And this is one example of, I don't know how well you can see this. This is a fly through the hippocampus. So this is the area CA1 at the top. So this is where we're going. We're going down through CA1. So my cursor is indicating where we are about. At one point, you see an increase in fluorescence, means that we get to the molecular layer of the dentate. This is it. We're at the molecular layer of the dentate. And now we're going to see deeper. You're going to see the dentogranule cells, and in the end, the mossy cells firing away. And we're about 700 microns. So you can image really deep. You can image big parts of the brain with this approach. The Grin lenses, there's different approaches to implanting Grin lenses. And if you have these tiny Grin lenses, you can either implant them alone, or meaning just the Grin lens itself, or you have these systems that have sleeves or something that avoids it.
What's a Grin lens?
We're going, next slide. Yeah, this is that's true. It wasn't a very good idea. I realize now. They should be the other way around. So Grin lenses. So Grin lenses, what they are basically lenses. They're light guides or lenses. They're rod-shaped lenses with a radially varying refractive index. So what they do is that they have their refractive index. Whereas a regular lens, you imagine a regular lens being something like this, and you basically have your focusing effect is because you have your rays go through different lengths of material, different path lengths of material with a different refractive index, which is glass. Here, these lenses bend the light by having a different refractive index in the center from the periphery. And you see here, this is the refractive index, a plot of the refractive index, and is approximately parabolic. It's not quite parabolic, but it's approximately parabolic, and it's actually quite a lot of technology that goes into this. So they dope the glass with incorporation of silver, lithium, or thallium ions, depending on the process. And they make these lenses, which are basically, this is one example, that it's basically a rod of glass, a tiny rod of glass. And they can be 1 millimeter. In this case, this is 0.5 millimeters. If you ever lose one of these in the lab, it's hell, because you're going to be like crawling through the linoleum and like just to find this, because these are really tiny things. And that's why they're great for using the brain. The drawback is, of course, you have a small field of view, and wherever you implant it, you're stuck there. So also, Grin lenses have a relatively, Claire. Let me see.
I have a question, just, it's more general, but in all of these images, you have the objective is like perfectly perpendicular to the lens or the cover slip. Is that necessary? Do you
It's
have to
recommended.
turn the animal, say if you're imaging like there?
It's recommendable, and the reason is, you know, I won't go into the details here, but there's simple ray tracing. You can figure out that if you're not aligned perpendicular to your window, even in the simplest example of the window, you will actually introduce a small aberration. So your rays coming in from your objective will not all be focused on the same spot. So that degrades your quality of imaging. Yeah, so it is important. And there's ways to mitigate this. So you can actually have, we have a system in our microscope where we shine a little laser diode along the optic axis of the microscope. It's a little dot that is projected, and it's back reflected from the cover slip. And you project this back into the a little camera, and then you can center this. You basically use a bullseye to align the animal very precisely with these micromatic screws. So that was actually an excellent question. Yeah. Did I answer that? So it is important, and there's ways to compensate for it and align the animal appropriately. In some systems, you actually align the microscope entirely around the animal.
Yeah, that answers my question.
So then drawbacks. So these can be used, these are basically relay lenses. So you can have, they relay whatever's underneath the, whatever's underneath the lens at a certain working distance, they relay it up. And you can either use these with 2-photon microscopes or with miniscopes, which are going to be these little implantable microscopes that we're going to be talking about. And if these are small enough, sorry, I don't know why. Okay, if these are small enough, they can be implanted, and there's all sorts of technologies for implanting them. And you can go quite deep into the brain, and you know, people say, well, you know, it's a very long rod of glass, but it's only 0.5 millimeters, so it's kind of the equivalent of having an electrode in there or something like that. Finally, large windows are crystal skull. And crystal skull is this, and there's different approaches to this. This one is from the Schnitzel lab at Stanford. And it's an approach to make these very, very large craniotomies that allow you to really, and this is something that is really one of the next frontiers in imaging in the cortex, is imaging different cortical areas simultaneously and being able to look at the propagation of information between different areas of the brain. And you can have these enormous windows that keep transparent, and it's really beautiful, beautiful stuff. And these are perfect for using with these very large microscopes that are called the meniscopes. And I'm going to talk a little bit about that in a while, but they're just really good for that. So inflammation and side effects of imaging. So imaging techniques are minimally invasive, but they obviously have detrimental effects even when, no matter what you do. Just a laser shining in the brain creates, heats up the tissue. Inflammation in the surgery area is best to image acutely or after 2 to 3 weeks when the inflammation subsides. You have the bleaching of the fluorophore, the heating up of the tissue and so forth also creates some effect. So what you need to do, well, controls. So control for your behavior, make sure that your behavior is still there when your mice have been through this imaging. Antibody stains for IBA1, for example. Morphology of the cells, is it unchanged? The spines and so forth. So these are all things that you need to do. And there's actually a paper showing that just implanting a window actually cools the temperature of the brain, because it turns out your skull is better at, so you can have all sorts of different, and whenever you image, you actually you can have the heating up and a heating effect from the laser, but you can also, because you have the water and the objective, if you don't heat up your objective, you will have a cooling of this particular area of the brain, meaning that maybe the metabolism is completely changed there. This is something to keep in mind. There are papers that have looked at brain heating induced by near-infrared lasers that you use for multifocal microscopy. And you can actually, if you use too much power, and this will be something like over 100 milliwatt over a 1 millimeter square area, then you can have this increase of 1.8 Celsius. And there's papers that estimated this and estimated what damage it could make. So these papers are available, and this is what you should use for estimating how much power after your objective you should be using. And remember, it's always about measuring power at the objective, and it's always about what kind of what is the volume where you'll have this laser power concentrated in the brain. Okay, another mystery figure. What is this? Any guesses? So I'll tell you, it's a dentate gyrus. So I'll tell you more. DCX is a marker for newborn neurons.
Are we seeing neurogenesis?
Yeah, you see neurogenesis in the gyrus. So you see that here 2 days, 1 week, 4 weeks. What's happening here is that you have a contralateral control and a virus injected side of the brain. The virus injected is actually an empty virus. And it's not expressing anything. It's not expressing GCAMP. It's not expressing anything. The virus only has it's basically the capsid with the ITRs of the AAV. And what we found is that these cells die if they're infected by AAVs. And in fact, in culture, it's not an immune system response. It's not inflammation. You can replicate this in culture. You can replicate it by simply electroporating the ITRs of an AAV into these cells, and they die. So always be on the lookout for possible side effects. So even AAV-based vectors, which are usually quite innocuous, can have toxicity in some systems. In our case, for example, they completely wipe out adult neurogenesis, and they do so permanently. Most of these points were made by Luke in his lecture. But long-term Cree overexpression, for example, when you use a virus to express Cree, can be problematic. Mixing AAVs is not a good way to express two constructs very often. You get either one or the other expressed. And the viral expression, and this is a comment I made. I saw that Luke actually put this in the comments for the lecture, for the last lecture. Viral expression of calcium sensors can lead to heterogeneities in the intracellular concentration, meaning your intracellular concentration of GCAMP will not be the same in every cell, which means that your calcium responses, your fluorescent responses may vary. Remember, GCAMP is a calcium buffer as well as a calcium sensor. And as such, GCAMP can actually have effects in the brain that you're not thinking about. Okay, so what kind of imaging do people do? So how are we doing on time? Okay, we don't have a lot of time, but I think this is a kind of more general part. So imaging morphology, a lot of applications in neuroscience are imaging spines, and you can image spines over many days, sometimes even over 3 months or more. And it's a big thing in neuroscience because obviously figuring out how much of synaptic turnover there is very important. You can also image axon and dendritic morphology, axon development, remodeling. Can image microglia migration, for example, inflammation, morphology. This is an example of spine imaging, axon imaging. What's really important for this kind of imaging, morphology imaging, oh yeah, so then again, another example is the dendrites, right, where you see these day in, day out. Morphology imaging is usually done under anesthesia because it's not really a fast process. You do not need to have the mouse anesthetized unless you think that the anesthesia will have some sort of side effect, which you may well. You're imaging small structures, spines, for example, and sometimes it can be difficult to image because your mouse is alive, so it has a pulse. And the pulse actually is enough to make slight fluctuations in the brain every time the heart beats. So some people actually sync the scanning of the microscope to the pulse of the animal. And in this way, you avoid scanning, you avoid your image moving whenever you're scanning. So you want to, whenever you do this imaging of morphology, you want to use bright bleaching resistant fluorophores, say a GFP or a Venus or YFP are among the best. So as stable as possible. If you're imaging spines, you want to use cytosolic fluorescent proteins. If you're using axons, it's best to use membrane bound. And sometimes it's very, I don't know if you talked about this, Luke, but in nucleoelectroporation, if you want to go very early in development, sometimes the best way to do this is to electroporate a construct in utero. So you take the embryos out, you inject, you apply a pulse, you put the embryos back into the mom gives birth, and then you can start studying immediately after birth. You can start imaging. Another very common form of imaging is imaging blood flow. So this is pioneered by David Kleinfeld and Winfried Denk. You inject a dye into the vasculature, either the tail vein or the eye, the sinuses behind the eye, and then you go in and you home in on one of these little capillaries, and you do a line scan. A line scan is basically just using your fast axis of your mirror and just using it over one particular line. So you're going extremely fast because you're not making the full image. You're only scanning that one point back and forth. And then because you have a dye, usually a dextrin dye that doesn't permeate, it doesn't leave the vasculature, it's heavy, it has a certain molecular weight, it doesn't leak out, at least not easily. So what you do is that you start seeing, and here in some of these, you see that these little black lines. So this is what it looks like. So this is 100 milliseconds. So this is a time image of a line scan. And you see you have these, the white stuff here, the gray stuff is actually the blood. And the black lines are red blood cells crossing through. And what this allows you to calculate is from this angle and from the time, it allows you to calculate how many red blood cells per second are flowing. So this is the way, and here you see that these kinds of measurements. You know, in this case, Riccardo Mostani, who authored this work, he did a stroke in this animal. You see immediately here in the stroke, you see how perfusion has changed. It's a really beautiful visual way to see how many red blood cells were going through before the stroke and after the stroke, right? So yeah, and this is how the actual video, I don't know how well the video, these are too big, the caliber of these capillaries. This is actually an experiment we did where we put brain organoids inside the mice, and we use the mice as a little bit, as crude as it sounds, as an incubator. And then we had these organoids that were derived from embryonic stem cells, and they're human brain organoids. They were perfused by the mouse's vasculature, and you can image this. This is actually a little bit out of place. So the other type of imaging, and this is kind of the part that you're waiting for, is calcium imaging activity. Luke talked about this. So we have a bunch of tools for imaging activity. The most successful ones are calcium indicators. There's intensiometric dyes, which are the most common, like GCamp. Actually, if you have FRET-based sensors, and the FRET-based sensors have both the donor and the acceptor in the same molecule, and you're only expressing, so the ratio of both is constant, you can actually do ratiometric images and get absolute calcium concentrations even in vivo. It's not something that is done very often, but it is. So Luke already covered most of this. So you have the latest and bestest GCamps are the GCamp 7s. So you have the slow, which is very sensitive, the fast, that allows you to go really fast. Then you have the 7B, higher baseline, is very good for imaging, for example, axons, and so forth. It's always good to validate calcium sensor responses in your system. So here, for example, is an example where I was patching a cell, I had the pipette here, and I was able to get the calcium imaging and the recording. And then you can see how many, in different setups at different speeds of imaging, you can see how many of your single spikes are detected, how many of your doublets are detected, and if you have 3 or more action potentials, how much you can detect. Now, the most common method for relating electrophysiology to to calcium imaging is deconvolution. And this is Ken Harris. I don't know why, when I did the screen capture, this thing was here. This is from Ken Harris's lab. And it turns out that people don't do that because in many areas you cannot patch clamp an image at the same time. You can do it perhaps in a slice, but you can't patch clamp the hypothalamus and image at the same time with a Grin lens. That's impossible, right? So you'll never be able to establish this ground truth of what the relationship between calcium and electrophysiology is. And these guys actually say, well, it probably doesn't matter. So they did all these simulations, and they say, you know, this is spike deconvolution is pretty robust. And of course, we like to believe that, but, you know, and we do. I mean, I can't do that in the hippocampus. I can't patch. I can do that in the cortex. So, of course, the types of imaging that most people do with activity is really imaging activity of cells. Here you see in the visual cortex, a lot of cells flashing. This is the most common form of activity imaging. Next week, I'll try to talk a little bit about all optic interrogation of circuits, maybe like the ability to use channel-Rhodopsin and imaging simultaneously. The other thing you can do is image activity directly at synapses. So you can actually, and this is work by the Connor's lab, you can actually image calcium signals from individual synapses, which is just really amazing. And in this paper that they had in 2010, they actually showed that neurons in the visual cortex, individual synapses can have, they can be orientation selective, which is really, really cool. And if you guys have covered that in your neuroscience classes, you know what orientation selectivity is in V1, sensory systems. So this is a really cool example of the power of in vivo imaging. Then conversely, you can image the spines, but you can also image the axons. And this is work by Alberto Cruz Martin, imaging the selectivity of thalamocortical axons in the visual cortex and showing that these axons are already have a certain selectivity. So I said I was going to talk to you about the mesoscope. This is one of the big, so you saw that huge crystal skull window. So this is one of the big new things, I think now maybe about 4 or 5 years old, but it's this ability to huge. So this is the objective. You see this gigantic thing. You have no, this thing takes up a whole room. It's like this huge gantry with a microscope attached to it. And then there's a tiny mouse somewhere in there. But you know, your microscope is just gigantic. But what this allows you to do is to image. So look at the scale bar. This is 1 millimeter. And you see that this scale bar is changing. You're imaging just so you can zoom in into individual cells. You can zoom back out. You can go between one area of the brain and another. And you can cover millimeters in the brain. And this is kind of one of the latest developments in the field, being able to do these recordings with a single objective, jump from one part of the brain to the other part of the brain really, really fast. And I mean, this video is really impressive. You can look it up if you want. I don't know how well it reproduces in your Zoom. So of course, Luke also talked to you about imaging other probes and sensors, neurotransmitter release, for example, like the glue sniffers and D-light, intracellular signaling like cyclin MP, and even RNA transcription and transport, for example, Rob Singer at Einstein is doing that. Of course, that you know, a big holy grail of in vivo imaging is voltage imaging, and one of the kind of latest additions to our toolbox is Voltron, and there's others too. So these are all these voltage sensors. Imaging voltage is a lot harder than imaging calcium, because you need to, voltage is something that you can only measure at the membrane, as Luke explained, and it's something that is much it's changing at a much faster time scales than calcium. So these Voltron probes, they have these membrane proteins that are sensitive to voltage, and they use these halo tags and these Janelia fluo tags that are highly resistant to photon bleaching. And these guys, this is a really, you cannot image for very long, apparently. There's a few issues, but it's really, really impressive how far we've gone. So this is, again, visual system, and you can see individual spikes, and you can go, we're getting to the point where we can start doing electrophysiology in vivo with imaging. Voltron, we gave a journal club on this recently, and it's really sensitive. It can really capture action potential timing, even in fast spikers, and subthreshold activity, but it still requires a lot of power for imaging. It's single photon imaging. You need a fast camera, very quick imaging. It limits this to 4 to 5 cells that you can image. And still bleach is relatively fast compared to calcium imaging. And the fluorescent change is negative, which is not very good because your movement artifacts are also negative, right? So you always want, whenever you have a sensor, it may sound like a silly thing, but you always want it to go up instead of down. And there's other, this is Archon from Edward Bowen and Trehan. There's other types of sensors out there. Now, in the few minutes that I have left, I'm going to be talking a little bit about head-mounted miniscopes. Head-mounted miniscopes has been one of the great revolutions in the last few years. And the reason is you can do all sorts of, sorry, I skipped a couple of, you can do all sorts of behaviors in head-fixed mice. So obviously, in order to image in a microscope, in a 2-photon microscope, you need to fix the mice. So the mice are physically attached, and they need to be trained to engage in certain tasks, or they need to be restrained to look at sensory stimulation and so forth. And there's very different approaches. The very famous mouse and styrofoam ball. There's the treadmill approach. There's amazing stuff where mice actually voluntarily come up to the microscope. They walk up and they attach themselves to the microscope for a reward. And the lab of Mark Harnett at MIT just came out with this amazing navigation system where the mice are navigating in a little plate that is suspended, and it's almost like freely moving. They feel like it's freely moving, but they're actually in the same places. It's like this room that they're in that moves. So all of this is possible, but really sometimes it can be, this really limits your ability to do a bunch of different tasks and limits the movement of the mice. So what you want to do is have systems where the mice are more free to engage in different tasks, and head-mounted miniscopes are the answer to that partially. So there's these tiny sub 3 gram, and there's actually very many different versions out there. They were initially pioneered by Mark Schnitzer and other groups, Helmchen. I'm afraid I will forget if I start saying who they are. There's a lot of people collaborate, but Mark Schnitzer really did a bunch of these breakthroughs. And one of the most widely used systems comes out of his lab. And they're mounted for each imaging session. So you keep a grin lens in there usually, and you mount a miniscope. It has a little mount, a little plate, and you mount it every time you want to image a mouse. It can be single photon. Usually it's single photon. Now there's these 2-photon microscopes, miniscopes, and the mice are usually tethered. But there's now newer untethered versions that are completely wireless. So they're still they're still carrying this. I mean, for me, it would be the equivalent of carrying I don't know, like 15 pounds on my head, right? So it's still significant. It still restrains them to a certain extent. But no doubt, this has been a great improvement. It's also, they're relatively cheap. There's commercial sources like Inscopics, but there's also the UCLA Miniscope, which is an open source, and there's other open sources. So you can image mice, freely moving animals, easy to use. The do-it-yourself systems are really cheap. It's 5,000. This is much cheaper than a 2-photon microscope. The drawbacks is that they're single photon. They have lower resolution, lower signal to noise ratio, relatively small fields of view, and you're limited to in your working distance, and you're limited kind of where you land. Most systems are tethered, even though now you have wireless. Fewer collars, and actually the new 2-collar systems have just become available. And 3 grams is sometimes, even that can be, and now I think they have a 2.5 gram or something, but even that can be a little bit heavy. What's really amazing about these devices is how simple they are. They actually do use a CMOS sensor, so it's a camera sensor from a cell phone camera. They have these 3D printed bodies with just a relay lens. The emission filters, so the fluorescence filters, they're built in. They're these tiny, teeny emission filters in this box. They have the electronics built in, and then you have this base plate and the green lens that you have implanted. And this is all attached to the mouse itself. And you know, this is a recording, and this is taken from the miniscope, UCLA miniscope. See, this here is a mouse. This is a video camera of the mouse going back and forth in a linear track. And this is recording from cells. And this is what you see, this is the raw data. It's single photon. You see that it's a lot noisier, a lot cloudier than 2-photon imaging that I showed to you before. But with the right algorithms, and next week we're going to talk a little bit about that, you can still extract single cells. And so you can do a lot of stuff, like you can combine this with optogenetics. I'm running out of time, so I'll just say that now one of the great new things is this group in China, they have these hand-mounted microscopes. This used to be impossible to do, hand-mounted 2-photon microscopes. So that give you the 2-photon sectioning capabilities. And they have a bunch of innovations here. One of them is that they use these hollow core fibers. Remember that what I told you about the 2-photon lasers, they have these very short pulses and they disperse. So they basically cannot be sent usually over fiber optic. But there's these types of fiber that are newer that have a hollow core where they bounce around, and it turns out that you can actually send 2-photon titanium sapphire, very short femtosecond pulses down these fibers. They also have a low built-in scanner, because remember, if you go in 2-photon, you need to be, it's a scanning system. It's not a system where you illuminate, unlike the single photon miniscopes. They should have a little blue LED that illuminates everything underneath. These guys actually have a tiny miniature scanner. It's a MEMS scanner, which consists of a bunch of little mirrors that you actually deflect to form an image, to scan your image. And it's a really neat little thing. And you know, they have an example of a normal 2-photon microscope and a miniature implanted 2-photon microscope. And you see here, I think this is calcium imaging. I forget now. But I think when you see the flashes, you're actually seeing individual synapses, right? You see the individual spines here on the right. And this is really, when I first saw this, I was really very, very impressed. And this work kind of blew me away. And I know that we're just about to reach the end of the lecture, and this is kind of one of my final slides. So these guys, this is really amazing, and I expect that you'll see a lot more data coming from here. Now, the other thing that I wanted to tell you is that neuroscience doesn't stop at the brain, so there's a peripheral nervous system. There's a lot of stuff that is, and this is, you know, having a class is great because you can pontificate. At least you can pontificate until Luke and Anita cut me off. And you can talk about things that you just find cool. And one of the things that I find cool is that there's a lot of unexplored areas in the brain. Now people are starting to use optogenetics and imaging to look at stuff everywhere in the animal, the peripheral nervous systems. And you can stimulate and you can manipulate and image all of these nerves elsewhere. And even if you just look at work at Einstein from our neighbors like Paul Fournette over in Price, he has all this work about the importance of adrenergic innervation, both in prostate cancer and in controlling hematopoiesis in the bone marrow. And another example here, again, I just went to our neighbors on campus. This is work by David Antenberg and John Condilas. And they have this amazing window for 2 photon. This is not neuroscience at all, but this is, they implant this window in the lung. And I'm just blown away by this, because the lung, if you think the brain has movement, just think about the lung, right? With these mice inhaling and exhaling, and they have this beautiful work where they implant this long-term window, and they can see actually lung cancer metastasis going. For example, here you see these cells, they're making their way into metastasizing and infiltrating the bloodstream. And I think that there's a world of opportunity in things that you can do with these techniques. And this is only the beginning. And yeah, so that's it for me. I'll shut up now. And I want to know if you guys have any questions. Everyone's asleep.
I have a question.
Yeah.
So when the mouse are implanted with green lenses or all other kinds of head posting that are performed, is there how far away is the behavior of those mice versus the ones that are not implanted? Is it do they get like hyper, or like how far away?
Yeah, so this is something you need to that's why I had this slide where I said, you really do the controls. For example, we image it at the campus. Turns out that one well-known effect is that, for example, if you started blading CO1 and removing parts of CO1, you can get this hyperactivity. So you just put a mouse in an open field,
Mm-hmm.
and you start seeing that the mouse moves more. So there's all of these drawbacks sometimes. So there's stuff like that you need to control for. It will depend on what area of the brain you're imaging. It will depend on whether that's important for the behavior you're studying. But you're putting something into the brain. I mean, the same with putting an electrode into the brain too, right? I don't think I have anything really smart to say about that. I probably don't have a lot of smart things to say about anything, but especially about that, just do the controls, basically. You need to do the controls for your particular application, for your particular paradigm, behavior paradigm.
Right.
Claire, you had a question?
Yeah, so you were talking about like the administration of dexamethasone prior to the surgery as like a way to prevent swelling, and I was curious about there's this paper which is like in vivo optical clearing. It's on bioRxiv, and they basically like administer glycerol to the animals in their drinking water, which supposedly should reduce edema and somehow make the image clearer. Is it common to administer dexamethasone like long term to reduce swelling?
No, usually you don't do that long term. I mean, I've never done it long term. I don't know of anyone who does that. You can administer NSAIDs after surgery. In fact, many animal protocols have that also because of pain mitigation. The glycerol paper is a really interesting paper. I'd forgotten about that actually. Yeah, I don't know. I've never tried it.
Yeah,
and
I like their name for it too.
We do not administer dexamethasone continuously.
Right. Yeah. But yeah, I like
also,
their name.
I'm imagining that being a steroid, I'm not an MD, maybe Luke can chime in on that. Administering a steroid long term is probably not the best.
It's
Sorry,
fine
I
to
have
give
a question related to.
Sorry?
It's fine to give long-term steroids. I mean, as long as you taper them off. If you get them on, put them on too long and then immediately withdraw them, they go into, they have like adrenal insufficiency and all that. So you just have to taper them if they've been on for like a week.
That's good to know.
Sorry.
Sorry, Claire, I wanted to extend that question. What does glycerol do that improves imaging? Does it reduce swelling? What does it do?
In the paper, they're saying that it has like anti-inflammatory effects, but that's as far as I've read in it.
I remember reading that. I don't remember many of the details, but that's an interesting thing. I mean, I just don't remember. I think at the time I may have thought, oh, I need to try this, but it's like one of these many papers that he said, I need to try this, and then you're like too busy with other stuff.
Tiago, I have a question about the head-mounted 2 photons. So, do they have, I mean, so I remember the one that I saw, it had no mechanism to control the z-axis. Are there any ones that have that built in, or some way you can, like, you know, scan up and down?
I think so, but I think I saw that. But I thought it didn't have z-axis.
Well, I think it had a screw you can turn, but I mean, it didn't have like.
But nothing that is. Yes, there is. There is a paper by Naji using a Bessel beam to do scanning on to do Z scanning with these microscopes. Yeah, but I think that's kind of highly experimental. So, but there is a paper by Naji on that. That's all I know about it. Yeah. So I know that, yeah, they use a Bessel beam, and it sounded quite complex, but I haven't, you know, that's one of the things that I didn't talk about is the spatial light modulators and adaptive optics and stuff like that. Maybe I'll mention a little bit of that next week. All right. Sounds like no more questions. Should we stop recording then?
Bye, everyone.
All right. So now see you guys on Tuesday. Or is it Passover? I don't know.